3D printing technologies for 3D brain organoids, having functional neural networks and blood vessels. Introduction. The development of 3D printing technologies for creating brain organoids, with functional neural networks and blood vessels, represents a significant advancement in biomedical research. These technologies enable the fabrication of complex brain tissue models that closely mimic the natural brain environment, facilitating studies on neural development, disease modeling, and drug testing. On historical background, the evolution of 3D printing in biomedical research began with simple tissue models. Over time, advancements in bioprinting techniques have allowed for the creation of more complex structures, leading to the development of functional brain organoids. 1.2 Significance in biomedical research. 3D printed brain organoids provide unprecedented opportunities for studying brain development and pathology. They serve as models for understanding neurodevelopmental disorders, neurodegenerative diseases, and brain injuries. 1.3 Applications in drug testing and development. These organoids offer a more accurate platform for testing drug efficacy and toxicity, potentially reducing the reliance on animal models and improving the prediction of human responses to new therapeutics. 1.4 Impact on personalized medicine. The ability to create patient-specific brain organoids opens up new possibilities for personalized medicine, where treatments can be tailored to the individual's unique biological makeup. Fundamentals of 3D neural tissue printing. 3D neural tissue printing involves the use of bioprinting techniques to construct neural tissues with precise spatial arrangements. Biomaterials such as hydrogels, alginate, gelatin, collagen serve as scaffolds to support the growth and differentiation of neural cells. These scaffolds are designed to replicate the brain's extracellular matrix, creating a conducive environment for cell proliferation and network formation. 2.1 Bioprinting techniques. Various bioprinting methods, including inkjet, extrusion, and laser-assisted printing, are used to deposit cells and biomaterials layer by layer, forming the 3D structure of neural tissues. Inkjet bioprinting. Principle similar to traditional inkjet printing, droplets of bioink, a mixture of cells and biomaterials, are ejected onto a substrate to form a 3D structure. Advantages: high speed, cost-effective, and capable of printing multiple cell types. Limitations: limited resolution and potential damage to cells due to high shear stress. Extrusion bioprinting. Principle: continuous streams of bioink are extruded through a nozzle to build layers of tissue. Advantages: suitable for high-viscosity bioinks and larger-scale constructs. Limitations: lower resolution compared to other methods, and possible cell damage from pressure. Laser-assisted bioprinting. Principle: uses a laser to focus energy on a substrate, creating a high-pressure bubble that propels bioink droplets onto a surface. Advantages: high resolution and cell viability. Precise control over cell placement. Limitations: expensive and requires complex setup. Stereolithography (SLA). Principle: uses UV light to solidify layers of a photosensitive bioink, building up the 3D structure layer by layer. Advantages: high resolution and accuracy, good for complex structures. Limitations: limited to photosensitive materials, and potential cytotoxicity of UV light. Each technique has unique advantages and limitations, making them suitable for different applications in neural tissue engineering. Researchers often choose the method based on the specific requirements of the tissue they aim to construct. 2.2 Selection of biomaterials. The choice of biomaterials is crucial for the success of 3D printed neural tissues. Biomaterials serve as scaffolds that support the growth, differentiation, and organization of neural cells, providing a structural and biochemical environment that mimics the brain's extracellular matrix (ECM). The main criteria for selecting biomaterials include biocompatibility, mechanical properties, and the ability to promote cell adhesion, proliferation, and differentiation. Types of biomaterials. Hydrogels. Hydrogels are the most commonly used biomaterials in 3D bioprinting due to their high water content, which is similar to natural tissues. They provide a hydrated environment that supports cell viability and function. Common hydrogels include Alginate derived from seaweed, alginate, is widely used due to its biocompatibility and ease of gelation. It can be combined with other materials to improve cell adhesion. Gelatin, a derivative of collagen, gelatin is biocompatible and biodegradable, providing a supportive environment for cell growth. It can be modified to control its mechanical properties. Collagen, the main structural protein in the ECM. Collagen supports cell adhesion and differentiation. It can be used alone or combined with other materials to enhance its properties. Matrigel, a complex mixture of ECM proteins, matrigel promotes cell attachment and differentiation, making it ideal for neural tissue engineering. Synthetic polymers. Synthetic polymers are used for their tunable mechanical properties and degradation rates. They can be engineered to mimic the physical characteristics of natural tissues. Polyethylene glycol PEG hydrogels are customizable in terms of stiffness and degradation, making them suitable for various tissue engineering applications. Polylactic acid PLA is biodegradable and has good mechanical strength, often used for creating scaffold structures. Natural polymers. Natural polymers provide biological cues that enhance cell interaction and functionality. Chitosan, derived from chitin, chitosan, has antibacterial properties and promotes cell adhesion and proliferation. Hyaluronic acid, found in the ECM, hyaluronic acid supports cell migration and proliferation, contributing to tissue regeneration. Properties of biomaterials. Biocompatibility. Biocompatible materials do not elicit an immune response and support cell survival, proliferation, and differentiation. Ensuring biocompatibility is critical for maintaining cell viability. In 3D printed constructs. Mechanical properties. The mechanical properties of biomaterials, including stiffness and elasticity, should match those of the target tissue. For neural tissues, softer materials that mimic the brain's pliability are preferred. Adjusting the cross-linking density of hydrogels can fine-tune their mechanical properties. Degradation rate. The degradation rate of biomaterials should align with the rate of tissue regeneration. Materials that degrade too quickly may not provide sufficient support, while those that degrade too slowly may impede new tissue formation. Defunctionalization of biomaterials. Bioactive molecules. Incorporating bioactive molecules such as growth factors, peptides, and ECM proteins into biomaterials can enhance their functionality. These molecules promote cell adhesion, growth, and differentiation. Surface modification. Surface modification techniques, such as coating with ECM proteins or functionalizing with peptides, improve cell material interactions. This enhances the integration and functionality of the printed neural tissues. Challenges and innovations. Balancing properties. Achieving the optimal balance between mechanical strength, biocompatibility, and degradation rate remains a challenge. Innovations in biomaterial engineering are focused on creating hybrid materials that combine the best properties of natural and synthetic polymers. Customization for patient-specific applications. Developing customizable biomaterials that can be tailored to individual patient needs is an emerging area of research. This involves creating materials that can be precisely adjusted in terms of mechanical properties, degradation rates, and bioactive molecule incorporation. Enhancing vascularization. Improving the vascularization of 3D printed neural tissues is a critical area of innovation. Biomaterials that promote angiogenesis and integrate with vascular networks are being developed to enhance the viability and functionality of printed brain organoids. Future directions. Future research aims to develop advanced biomaterials that better replicate the complexity of natural brain tissues. This includes exploring new sources of natural polymers, engineering multifunctional synthetic polymers, and integrating smart materials that respond to environmental cues to enhance tissue development and functionality. 2.3 Cell sources and differentiation. In the realm of 3D neural tissue printing, selecting the appropriate cell sources and guiding their differentiation into functional neural tissues is crucial for the success of the process. Here, we delve into the types of cells used, their origins, and the methods employed to differentiate them into the required neural cell types. Pluripotent stem cells, including induced pluripotent stem cells, iPSCs, and embryonic stem cells, ESCs, are commonly used to generate neural cells for printing. These cells can differentiate into various neural cell types, forming complex neural networks. Types of cells used. Various cell types are used in 3D neural tissue printing, including pluripotent stem cells (PSCs). These include both embryonic stem cells, ESCs, and induced pluripotent stem cells, iPSCs. PSCs are highly versatile and can differentiate into any cell type, making them ideal for generating neural tissues. Neural stem cells (NSCs). These are multipotent cells specifically committed to generating neural lineages, including neurons, astrocytes, and oligodendrocytes. Primary neural cells, cells directly harvested from brain tissue. Although they closely resemble natural neural tissues, their availability and ethical sourcing are more challenging. New origin of cells. Embryonic stem cells (ESCs) derived from the inner cell mass of blastocysts. ESCs have the ability to differentiate into any cell type. However, their use raises ethical concerns. Induced pluripotent stem cells (iPSCs) generated by reprogramming adult somatic cells to a pluripotent state. iPSCs offer a more ethically viable alternative to ESCs. They can be derived from the patient's own cells, enabling personalized medicine applications. Neural stem cells (NSCs) typically isolated from fetal or adult brain tissue. NSCs are naturally predisposed to form neural tissues, reducing the complexity of differentiation protocols. Differentiation protocols. The process of differentiation involves guiding stem cells to develop into specific neural cell types through a series of steps. Key differentiation protocols include directed differentiation using specific growth factors and signaling molecules to direct the differentiation of PSCs into neural progenitors, and subsequently into neurons, astrocytes, and oligodendrocytes. For example, the addition of retinoic acid can promote the differentiation of iPSCs into motor neurons. Spontaneous differentiation, allowing PSCs to differentiate in a 3D culture environment without specific external cues. This method often leads to the formation of diverse cell types, mimicking early neural development. Co-culture systems combining PSCs or NSCs with other cell types to create a supportive environment that promotes neural differentiation. For instance, CO culturing with astrocytes can enhance the maturation and functional integration of neurons. Techniques for enhancing differentiation. Biochemical cues, growth factors such as brain-derived neurotrophic factor (BDNF) and nerve growth factor (NGF) play essential roles in promoting the survival and differentiation of neural cells. Mechanical cues, substrate stiffness and mechanical forces can influence cell differentiation. Using hydrogels with varying stiffness can mimic the brain's extracellular matrix and guide neural differentiation. Electrical stimulation, applying electrical fields can enhance the maturation and connectivity of neural networks, promoting synapse formation and functional integration. Challenges and considerations. Efficiency and consistency. Achieving high efficiency and consistency in differentiation protocols remains a challenge. Variability in cell lines and culture conditions can lead to inconsistent results. Ethical and regulatory issues. The use of ESCs poses ethical concerns. While the reprogramming techniques for iPSCs must meet regulatory standards to ensure safety and efficacy, long-term viability, ensuring the long-term survival and functionality of differentiated neural cells within 3D printed tissues is essential for their practical applications in research and therapy. By addressing these factors, researchers aim to optimize the use of cell sources and differentiation protocols in 3D neural tissue printing, advancing the field toward more reliable and functional brain organoids. 2.4 Structural and functional validation. Post-printing validation involves assessing the structural integrity and functional properties of the printed tissues. Techniques such as immunohistochemistry, electrophysiology, and live imaging are used to evaluate cell viability, differentiation, and network activity. Structural and functional validation is a crucial step in ensuring the success of 3D printed neural tissues. This process involves a series of techniques and methodologies to assess the integrity, viability, and functionality of the printed tissues. Below, we dive deeper into the various aspects of structural and functional validation. Structural validation. Microscopic analysis. Microscopic analysis, including light microscopy, confocal microscopy, and electron microscopy, is used to examine the microstructure of the printed tissues. This includes evaluating cell distribution, scaffold architecture, and the overall morphology of the neural tissues. Immunohistochemistry. Immunohistochemistry (IHC) involves staining the tissues with antibodies that target specific cellular markers. This technique helps identify different cell types, assess cell proliferation, and detect the presence of neural markers, such as NUN for neurons and GFAP for glial cells. Histological staining. Histological staining, using dyes like hematoxylin and eosin, H&E, provides detailed images of tissue structure. It highlights cellular organization, tissue integrity, and the presence of any structural abnormalities. Imaging techniques. Advanced 3D imaging techniques, such as MRI and CT scanning, offer a comprehensive view of the entire tissue construct. These methods help visualize the internal architecture, including the integration of vascular and neural networks. Functional validation. Electrophysiological studies. Electrophysiological studies, including patch clamp recordings and multi-electrode arrays, MEAs, are used to measure the electrical activity of the printed neural tissues. These techniques assess the functionality of neurons by recording action potentials, synaptic activity, and network oscillations. Calcium imaging. Calcium imaging involves using fluorescent indicators to visualize intracellular calcium fluxes, which are indicative of neuronal activity. This method allows real-time monitoring of neural network activity and connectivity. Functional assays. Functional assays, such as neurotransmitter release assays and synaptic plasticity tests, help evaluate the functional properties of synapses and the ability of neural networks to undergo plastic changes in response to stimuli. Behavioral assays. In more advanced applications, organoids can be implanted into animal models to study their integration and functional contribution to the host brain. Behavioral assays can then be used to assess the impact of the implanted tissue on cognitive and motor functions. Viability and longevity. Live dead staining. Live dead staining uses fluorescent dyes to distinguish between living and dead cells within the tissue. This assay provides a snapshot of cell viability shortly after printing and over extended culture periods. Metabolic activity assays. Metabolic activity assays, such as MTT and ATP assays, measure the metabolic activity of cells within the tissue, serving as an indirect indicator of cell viability and function. Long-term culture studies. Long-term culture studies monitor the survival, growth, and functionality of the printed tissues over weeks or months. These studies help assess the stability and longevity of the neural networks and vascular structures. Integration and interaction studies. Co-culture systems. Co-culture systems involve growing printed neural tissues with other cell types, such as astrocytes, microglia, or endothelial cells, to study cell-cell interactions and the impact on neural network formation and functionality. Integration with host tissue. For in vivo applications, the integration of 3D printed tissues with host tissue is assessed by implanting the organoids into animal models and observing their survival, integration, and functional contribution to the host brain. Advanced analytical techniques. Transcriptomic and proteomic analysis. Transcriptomic and proteomic analyses provide insights into the gene expression, profiles, and protein compositions of the printed tissues. These techniques help understand the molecular pathways involved in neural differentiation and network formation. Single-cell sequencing. Single-cell sequencing allows for the analysis of individual cells within the tissue, offering detailed information on cellular heterogeneity and the differentiation status of neural cells. Bioinformatics. Bioinformatics tools are used to analyze large datasets generated from transcriptomic, proteomic, and sequencing studies. These analyses provide comprehensive insights into the biological processes and pathways active in the printed tissues. Summary: structural and functional validation is essential for ensuring the quality and reliability of 3D printed neural tissues. By employing a combination of microscopic, electrophysiological, biochemical, and analytical techniques, researchers can thoroughly evaluate the structural integrity, cellular viability, and functional capabilities of these complex tissue constructs. This rigorous validation process is critical for advancing the applications of 3D printed brain organoids in research, drug testing, and therapeutic interventions. Advanced techniques for vascularization. The incorporation of functional blood vessels within 3D printed brain organoids is crucial for their viability and functionality. Techniques such as the use of HPSC-derived parasites and endothelial cells facilitate the formation of vascular networks. These cells can sprout and self-assemble into organized vascular structures, providing necessary nutrients and waste removal pathways for the organoids. 3.1 Vascular network formation. Creating a vascular network within organoids involves co-culturing neural cells with endothelial cells and parasites, which form blood vessels through processes like angiogenesis and vasculogenesis. Creating a functional vascular network within 3D printed brain organoids is a critical step in ensuring their viability and mimicking the natural brain environment. This process involves several sophisticated techniques and strategies. Co-culture of neural and vascular cells. Co-culturing neural cells with endothelial cells and parasites is essential for forming blood vessels. Endothelial cells line the interior surface of blood vessels, while parasites support and stabilize these structures. Together, they can create a network that mimics the brain's vasculature. Angiogenesis and vasculogenesis. Angiogenesis. This process involves the growth of new blood vessels from pre-existing ones. In the context of brain organoids, angiogenesis can be stimulated by growth factors such as VEGF, vascular endothelial growth factor, which encourages endothelial cells to sprout and form new vessels. Vasculogenesis. Unlike angiogenesis, vasculogenesis involves the formation of entirely new blood vessels from endothelial progenitor cells. This process is crucial for creating the initial vascular framework within the organoid. Bioprinting techniques. Layer-by-layer printing. This method involves printing neural and vascular cells in a precise, layered manner to form an integrated structure. Each layer is printed with specific patterns that guide the formation of vascular networks. Sacrificial bioprinting. This technique uses a temporary scaffold or sacrificial material that is later removed, leaving behind a network of hollow channels. These channels can then be lined with endothelial cells to form blood vessels. Use of growth factors and signaling molecules. Growth factors play a vital role in the development of vascular networks. Besides VEGF, other factors like FGF, fibroblast growth factor, and PDGF, platelet-derived growth factor, are used to promote the proliferation and differentiation of vascular cells. These molecules ensure that the vascular network grows in a controlled and organized manner. Scaffold materials for vascularization. Choosing the right scaffold material is crucial for supporting vascular network formation. Biomaterials such as fibrin, a natural protein involved in blood clotting, used to create supportive and biodegradable scaffolds. Collagen, a primary structural protein in the extracellular matrix, providing strength and support to tissues. Hyaluronic acid, a component of the extracellular matrix that supports cell migration and proliferation. Perfusion systems. To maintain and enhance the functionality of the vascular network, perfusion bioreactors are used. These systems simulate blood flow by continuously circulating nutrient-rich media through the vascular network, providing oxygen and nutrients while removing metabolic waste. This dynamic environment is essential for the maturation and longevity of the organoid. Integration with neural networks. The integration of vascular networks with neural tissues is essential for creating a functional brain organoid. This integration ensures that neural cells receive adequate nutrients and oxygen, which are crucial for their survival and function. Techniques to enhance this integration include gradient-based placement, positioning neural and vascular cells in gradients to promote natural interactions and integration. Bioactive molecules incorporating molecules that encourage neural vascular interactions and stability, challenges and future directions. Despite significant progress, several challenges remain in vascular network formation. Within brain organoids, scalability creating vascular networks in larger or more complex organoids. Functional maturation ensuring that these networks are fully functional and capable of supporting the organoid long-term. Replicating brain-specific vascular properties mimicking the unique characteristics of the brain's vasculature, such as the blood-brain barrier. Future research aims to overcome these challenges by developing more advanced bioprinting techniques, exploring novel biomaterials, and improving the biological cues that guide vascular network formation. 3.2 Biomaterials for vascularization. Biomaterials such as fibrin, collagen, and hyaluronic acid are used to support the growth of vascular cells. These materials provide a scaffold that mimics the natural environment of blood vessels. Types of biomaterials used for vascularization. Natural hydrogels. Natural hydrogels, such as collagen, fibrin, and gelatin, are commonly used for creating vascular networks. Due to their biocompatibility and ability to mimic the natural extracellular matrix ECM of tissues. Collagen is a major component of the ECM and supports cell adhesion, migration, and differentiation. It provides structural support and can be easily remodeled by cells. Fibrin gels are derived from fibrinogen and thrombin. They support cell migration and proliferation, and are commonly used in wound healing and tissue engineering applications. Gelatin is derived from collagen, and is often used as a scaffold material, due to its ability to form stable gels that support cell attachment and growth. Synthetic hydrogels. Synthetic hydrogels, such as polyethylene glycol PEG and polyvinyl alcohol PVA, offer tunable mechanical properties and degradation rates, allowing for precise control over the scaffold environment. Polyethylene glycol PEG hydrogels are biocompatible and can be functionalized with bioactive molecules to promote cell attachment and growth. They are highly tunable in terms of stiffness and degradation rates. Polyvinyl alcohol PVA hydrogels are known for their excellent mechanical properties and stability. They can be engineered to degrade at controlled rates and are often used in combination with other biomaterials. Functionalization of biomaterials. Growth factors. Incorporating growth factors such as vascular endothelial growth factor VEGF and basic fibroblast growth factor BFGF into the biomaterials can significantly enhance angiogenesis and the formation of functional blood vessels. Vascular endothelial growth factor VEGF is a key regulator of angiogenesis, stimulating the growth of new blood vessels from pre-existing ones. It is often incorporated into hydrogels to promote vascular network formation. Basic fibroblast growth factor BFGF supports the proliferation and differentiation of endothelial cells, and is used to enhance the stability and functionality of newly formed blood vessels. Peptide sequences. Bioactive peptide sequences, such as RGD, arginine glycine aspartic acid, can be incorporated into the biomaterials to enhance cell adhesion, migration, and proliferation. RGD peptide. The RGD sequence is a key cell adhesion motif found in many ECM proteins. It promotes the attachment and spreading of endothelial cells, facilitating the formation of vascular structures. Engineering biomaterials for controlled degradation. Enzymatically degradable hydrogels. Hydrogels can be engineered to degrade in response to specific enzymes produced by cells, allowing for the gradual release of encapsulated cells and growth factors. Matrix metalloproteinase MMP sensitive hydrogels. These hydrogels degrade in response to MMPs secreted by migrating cells, providing a dynamic environment that supports tissue remodeling and vascularization. Picohenry's sensitive hydrogels. pH-sensitive hydrogels degrade in response to changes in the local pH environment, which can be triggered by cellular activity or disease conditions. pH-responsive hydrogels. These hydrogels can be designed to degrade at specific pH levels, allowing for controlled release of therapeutic agents. and promoting tissue regeneration. Case studies of biomaterial applications in vascularization. Collagen-fibrin hybrid scaffolds. Researchers have developed hybrid scaffolds, combining collagen and fibrin to create robust and highly vascularized tissues. These scaffolds support the formation of stable and functional blood vessels, enhancing the survival and integration of implanted tissues. PEG-based hydrogels with VEGF. Studies have shown that PEG-based hydrogels, functionalized with VEGF, can significantly enhance vascularization in tissue-engineered constructs. These hydrogels provide a controlled release of VEGF, promoting the formation of mature and functional vascular networks. Future directions in biomaterial development. And smart biomaterials. The development of smart biomaterials that respond to environmental stimuli e.g., temperature, pH, and mechanical stress, holds great promise for dynamic and adaptive tissue engineering applications. Nanotechnology-enhanced biomaterials. Incorporating nanotechnology into biomaterial design can enhance the mechanical properties and bioactivity of scaffolds, providing new opportunities for creating highly functional and vascularized tissues. Summary. Biomaterials play a crucial role in the vascularization of 3D printed brain organoids. By selecting appropriate materials and incorporating bioactive factors, researchers can create complex and functional vascular networks that enhance the viability and functionality of printed tissues. Ongoing advancements in biomaterial science will continue to drive innovations in tissue engineering and regenerative medicine. 3.3 Perfusion bioreactors. Introduction to perfusion bioreactors. Perfusion bioreactors are sophisticated systems designed to simulate the dynamic environment of blood flow within biological tissues. They play a crucial role in the development of 3D printed brain organoids by ensuring the delivery of essential nutrients and oxygen, while removing metabolic waste products. This continuous flow system mimics the physiological conditions of the brain, enhancing the viability and functionality of the organoids. Mechanisms of perfusion bioreactors. Perfusion bioreactors operate by pumping culture medium through the bioprinted tissue constructs. The medium contains nutrients, growth factors, and oxygen necessary for cell survival and proliferation. The flow rate and pressure can be precisely controlled to mimic the natural blood flow within brain tissues. Key components of a perfusion bioreactor include: pump system regulates the flow of culture medium, reservoir holds the culture medium, tubing and channels direct the flow through the tissue constructs, chamber houses the bioprinted tissue, and facilitates exchange between the tissue and medium. The advantages of using perfusion bioreactors. Perfusion bioreactors offer several advantages over static culture methods. Enhanced nutrient delivery. Continuous flow ensures a steady supply of nutrients and oxygen. Waste removal. Efficient removal of metabolic waste prevents toxic accumulation. Improved cell viability. Better mimicking of physiological conditions leads to higher cell survival rates. Tissue maturation. Dynamic flow promotes the maturation of tissue constructs, enhancing their functionality. Applications in 3D printed brain organoids. In the context of 3D printed brain organoids, perfusion bioreactors are used to support vascularized tissues. Perfusion systems are essential for maintaining the viability of vascular networks within organoids, enhance neural network formation. Dynamic flow conditions promote the differentiation and connectivity of neural cells. Long-term cultivation allow for extended culture periods, enabling the study of long-term developmental processes and disease progression. Case studies and examples. Several studies have demonstrated the effectiveness of perfusion bioreactors in enhancing the development and functionality of 3D printed brain organoids. Harvard University researchers developed a perfusion system that significantly improved the viability and complexity of vascularized brain organoids. University of California, San Diego. A study utilized perfusion bioreactors to maintain functional neural networks within 3D printed brain tissues, enabling detailed studies of neurodevelopmental disorders, challenges and future directions. While perfusion bioreactors offer many benefits, they also present certain challenges. Complexity and cost. The design, and operation of perfusion systems can be complex and expensive. Optimization, fine-tuning flow rates and pressure to match the specific needs of different tissue types requires careful optimization. Integration with bioprinting, ensuring seamless integration of bioprinting processes with perfusion systems is critical for achieving consistent results. Future research is focused on developing more user-friendly and cost-effective perfusion bioreactors, as well as exploring their integration with advanced bioprinting techniques and materials. This will further enhance the potential of 3D printed brain organoids for biomedical research and therapeutic applications. 3.4 Challenges and Solutions. The incorporation of functional blood vessels within 3D printed brain organoids presents several challenges. Addressing these issues is critical for the successful development and application of these advanced tissue models. Below are some key challenges and potential solutions. One of the main challenges in vascularization is achieving the correct spatial organization and integration of blood vessels with neural tissues. Advances in bioprinting and biofabrication techniques are addressing these challenges, improving the complexity and functionality of printed brain organoids. Challenges. Spatial organization. One of the primary challenges is achieving the correct spatial organization of vascular networks within the organoids. The intricate and dense network of blood vessels in the brain is difficult to replicate using current bioprinting techniques. Integration with neural tissues. Ensuring that the vascular networks integrate seamlessly with neural tissues is another significant challenge. The blood vessels must be precisely aligned with the neural structures to provide adequate nutrients and waste removal without disrupting neural network formation. Vascular maturation. The maturation of printed vascular networks into fully functional blood vessels, capable of supporting long-term tissue viability, is a complex process. These vessels must develop properties such as permeability, stability, and the ability to respond to physiological signals. Biocompatibility and immune response. Biocompatibility of the materials used for vascularization and the potential immune response are critical considerations. Materials and cells used in bioprinting must not provoke adverse immune reactions that could compromise the integrity and functionality of the organoid. Perfusion and nutrient supply. Effective perfusion, which involves the flow of nutrients and oxygen through the vascular network, is essential for the survival and function of the organoids. Achieving and maintaining efficient perfusion is challenging due to the complexity of replicating natural blood flow dynamics. Solutions. Advanced bioprinting techniques. The use of advanced bioprinting techniques, such as multi-material and high-resolution bioprinting, can improve the spatial precision of vascular networks. These techniques allow for the precise placement of cells and biomaterials, enhancing the fidelity of the printed structures. Co-culture systems. Implementing co-culture systems where neural cells and vascular cells are grown together can promote natural integration. Co-culturing endothelial cells with neural progenitors encourages the formation of vascular networks that are better aligned with neural tissues. Growth factors and signaling molecules. Incorporating growth factors and signaling molecules that promote vascularization can enhance the maturation and functionality of the blood vessels. Factors such as VEGF, vascular endothelial growth factor, and PDGF, platelet-derived growth factor, play crucial roles in angiogenesis and vessel stability. Scaffold design and biomaterials. Using biocompatible scaffolds designed to mimic the extracellular matrix of both neural and vascular tissues can improve integration and maturation. Materials like gelatin, collagen, and fibrin provide a supportive environment for both cell types. Perfusion bioreactors. Employing perfusion bioreactors that simulate blood flow can enhance nutrient and oxygen delivery throughout the organoid. These bioreactors help maintain a dynamic environment that supports the long-term viability and functionality of the vascularized tissues. Immunomodulatory strategies. Developing immunomodulatory strategies, such as using immunoprivileged cells or designing materials that evade immune detection, can mitigate immune responses. This approach ensures the stability and longevity of the printed tissues. Case studies. Harvard University's vascularized organoids. Researchers at Harvard University have successfully used a combination of endothelial cells and parasites to create vascularized brain organoids. They employed a perfusion bioreactor system that allowed for the continuous flow of culture medium, significantly enhancing the viability and functionality of the organoids. University of California, San Diego's approach. Scientists at UC San Diego developed a method using bioprinting and microfluidic systems to create intricate vascular networks within brain organoids. This technique improved the perfusion and overall health of the tissues, demonstrating the potential for scalable production of functional organoids. Future directions. Continued research is focused on refining these techniques and overcoming existing limitations. Efforts include developing more sophisticated bioprinting platforms, exploring novel biomaterials, and enhancing the understanding of vascular and neural tissue interactions. The goal is to achieve seamless integration and long-term functionality of vascularized brain organoids, ultimately advancing their applications in research and medicine. Integration of neural networks. Researchers have developed methods to print neural tissues with defined cell types and spatial arrangements, enabling the creation of complex neural circuits. These printed tissues exhibit electrical activity and can respond to stimuli, similar to natural brain tissue. This capability is essential for studying neural connectivity and functionality in vitro. 4.1 Neural circuit design. Designing neural circuits involves arranging different types of neurons and glial cells in specific patterns to mimic the organization of brain regions. Computational modeling aids in predicting the optimal layouts for functional networks. Designing neural circuits for 3D printed brain organoids involves several critical steps and considerations to ensure the creation of functional and accurate models of brain tissue. These steps include defining the types of neurons and glial cells to be used, determining their spatial arrangement, and employing computational models to optimize the design. Below is a detailed breakdown of the neural circuit design process and selection of neural cell types. Neural circuits in the brain comprise various types of neurons and glial cells, each with specific functions. Different types of neurons, such as excitatory, inhibitory, and modulatory neurons, are selected based on the desired circuit functionality. Each neuron type has unique properties and connectivity patterns. Glial cells, astrocytes, oligodendrocytes, and microglia are essential for maintaining homeostasis, providing support and modulating synaptic activity. 2. Spatial arrangement of cells. The spatial arrangement of neurons and glial cells is crucial for replicating the structure and function of natural neural circuits. Layered structures in the cortex. Neurons are organized into distinct layers. Mimicking this layered structure is vital for creating functional cortical tissues. Region-specific patterns. Different brain regions have unique cellular architectures. For instance, the hippocampus has a distinct organization of pyramidal cells and interneurons critical for its function. Computational modeling and simulation. Computational tools play a significant role in designing neural circuits. Modeling neural networks. Computational models help predict how different configurations of neurons and synapses will affect network functionality. These models can simulate electrical activity and signal propagation. Optimization algorithms. Algorithms optimize the arrangement of cells to achieve the desired network behavior, such as specific patterns of electrical activity or synaptic connectivity. Printing techniques for neural circuits. Advanced bioprinting techniques are used to fabricate the designed neural circuits. Multi-material printing. Different cell types and biomaterials are printed simultaneously to create heterogeneous tissue structures. High-resolution printing techniques such as digital light processing, DLP, and two-photon polymerization allow for high-precision placement of cells and extracellular matrix components. Post-printing maturation. After printing, neural circuits require a maturation period, during which they develop functional synapses and networks. Growth factors. Adding growth factors and other bioactive molecules to the culture medium promotes cell differentiation and network formation. Electrical stimulation. Applying electrical stimulation can enhance synaptic connectivity and functional maturation. mimicking the natural activity-dependent development of neural circuits. Functional validation and testing. Validating the functionality of printed neural circuits involves several techniques. Electrophysiology techniques such as patch clamp recording and multi-electrode arrays (MEAs) measure the electrical properties of neurons and network activity. Imaging. Advanced imaging techniques, including calcium imaging and fluorescence microscopy, visualize neural activity and synaptic connections. Behavioral assays. For certain applications, printed neural circuits can be integrated into animal models to assess their functional integration and impact on behavior. Case studies and examples. Several research groups have demonstrated successful neural circuit designs. Cortical networks. Researchers have printed cortical-like structures that exhibit layered organization and functional connectivity, mimicking the natural cortex. Hippocampal circuit studies have replicated hippocampal circuits with functional properties similar to those of natural hippocampal tissue, enabling studies on memory and learning. Challenges and future directions. Designing and printing functional neural circuits face several challenges. Complexity. Replicating the intricate architecture and connectivity of natural neural circuits is technically challenging. Long-term viability. Ensuring the long-term viability and functionality of printed neural circuits remains a significant hurdle. Integration with vascular networks. Integrating neural circuits with vascular networks to provide adequate nutrition and waste removal is crucial for maintaining tissue health. Future research aims to overcome these challenges by developing more sophisticated printing techniques, exploring new biomaterials, and enhancing computational models for better predictive accuracy. By addressing these aspects, researchers aim to create more accurate and functional models of brain tissue, advancing our understanding of neural development, disease mechanisms, and potential therapeutic strategies. 4.2 Functional maturation. After printing, neural tissues undergo a maturation period, during which they develop functional synapses and networks. Factors such as growth factors, electrical stimulation, and mechanical cues are used to enhance this process. Functional maturation of 3D printed neural tissues is a critical phase in the development of brain organoids, where the cells transition from a preliminary state to forming functional synaptic networks that mimic natural brain activity. This process is essential for ensuring that the printed tissues can accurately model neural function and respond to stimuli in a manner similar to actual brain tissue. Growth factors and chemical cues. Growth factors such as brain-derived neurotrophic factor BDNF, nerve growth factor NGF, and glial cell line-derived neurotrophic factor GDNF play crucial roles in the differentiation and maturation of neural cells. These factors support the survival, growth, and differentiation of neurons, and are often included in the culture medium to promote functional maturation. Electrical stimulation. Applying electrical stimulation to developing neural tissues can enhance synaptic development and network formation. This technique mimics the natural electrical activity in the brain, encouraging the formation of functional synapses and improving overall tissue functionality. Electrical stimulation protocols vary, but typically involve applying controlled electrical pulses at specific frequencies and durations. Mechanical cues. Mechanical cues, such as substrate stiffness and microfluidic shear stress, can significantly influence neural differentiation and maturation. The mechanical environment of the culture system can be tuned to mimic the physical properties of the brain, promoting more natural tissue development. Techniques such as microfabrication and soft lithography are used to create these tailored environments. Synaptic development. Synaptic development is a key indicator of functional maturation. The formation of synapses, where neurons connect and communicate, is monitored through various methods such as immunohistochemistry, which labels synaptic proteins like synaptophysin and postsynaptic density protein 95PSD95. This allows researchers to visualize and quantify synaptic density and maturation. Electrophysiological activity. Electrophysiological techniques, including patch clamp recording and multi-electrode arrays, MEAs, are used to measure the electrical activity of the neural networks. These methods provide insights into the functional properties of the tissues, such as action potential generation, synaptic transmission, and network oscillations. The presence of spontaneous or evoked electrical activity is a hallmark of mature, functional neural networks. Calcium imaging. Calcium imaging is another powerful tool for studying functional maturation. This technique uses fluorescent indicators to track calcium fluxes within neurons, which correlate with neural activity. Changes in intracellular calcium levels during synaptic activity can be visualized in real time, providing a dynamic view of network function and maturation. Time course of maturation. The time course of maturation varies depending on the cell types used, the complexity of the tissue, and the specific culture conditions. Typically, it takes several weeks to months for 3D printed neural tissues to reach full functional maturity. Regular monitoring through the aforementioned techniques helps ensure that the tissues are developing appropriately and achieving the desired level of functionality. Optimization strategies. Optimizing the conditions for functional maturation involves fine-tuning various parameters, including the concentration of growth factors, the frequency and intensity of electrical stimulation, and the mechanical properties of the culture environment. Researchers often employ a combination of experimental approaches and computational modeling to identify the optimal conditions for promoting maturation. Challenges and future directions. Achieving complete functional maturation in 3D printed neural tissues remains challenging. Current efforts are focused on improving the reproducibility and efficiency of the maturation process. Future directions include the development of more sophisticated bioreactors that can better replicate the in vivo environment, as well as the integration of advanced imaging and monitoring technologies to provide real-time feedback during the maturation process. By addressing these aspects, researchers can enhance the functional maturation of 3D printed neural tissues, making them more reliable and effective models for studying brain function and disease. 4.3 Electrophysiological characterization. Techniques like patch clamp recording, multi-electrode arrays, MEAs, and calcium imaging are employed to study the electrical properties and activity of the printed neural networks. These methods help in understanding how the networks process and transmit information. Electrophysiological characterization is a critical component in evaluating the functionality of 3D printed neural networks within brain organoids. This process involves various techniques to measure and analyze the electrical activity of neural cells, providing insights into the connectivity and functionality of the printed tissues. Here, we explore the key methods used in electrophysiological characterization and their significance in the study of 3D printed brain organoids. Patch clamp recording. Patch clamp recording is a gold standard technique in electrophysiology, used to measure the ionic currents that flow through individual ion channels on the cell membrane. This method allows researchers to measure action potentials by isolating a small patch of the neural cell membrane. Researchers can record the action potentials that signify neural firing. Investigate ion channel function. Patch clamp can reveal the properties of various ion channels, crucial for understanding neural excitability and signaling. Multi-electrode arrays MEAs. Multi-electrode arrays MEAs are used to record electrical activity from multiple sites within a neural tissue simultaneously. Key features of MEAs include high-throughput analysis. MEAs can monitor the activity of large networks of neurons over extended periods. Network connectivity studies. They help in understanding how neurons within the organoid connect and communicate, providing data on network synchronization and signal propagation. Calcium imaging. Calcium imaging is a technique that uses fluorescent indicators to visualize calcium ion fluxes within cells, which are proxies for neural activity. This method offers several advantages. Real-time monitoring. Calcium imaging allows for the real-time observation of neural activity across the entire tissue. Activity mapping. It helps in creating detailed maps of active neural circuits within the organoid, revealing patterns of connectivity and activity. Or optogenetics. Optogenetics involves the use of light to control cells that have been genetically modified to express light-sensitive ion channels. This technique is particularly useful for precise stimulation. Optogenetics can precisely control the activity of specific neurons within the organoid, allowing for targeted studies of neural circuits. Functional analysis by selectively activating or inhibiting neurons. Researchers can study the effects on the overall network activity and behavior. Electrophysiological data analysis. Analyzing electrophysiological data involves several computational tools and techniques. To extract meaningful information, key aspects include spike sorting, identifying and categorizing individual spikes from recorded signals to determine the firing patterns of neurons. Network dynamics, analyzing the temporal and spatial dynamics of neural activity to understand how information is processed within the network. Correlation analysis, assessing the correlation between activities of different neurons to infer functional connectivity and network organization. Applications in disease modeling and drug testing. Electrophysiological characterization plays a pivotal role in using 3D printed brain organoids for disease modeling and drug testing. Disease phenotyping, by comparing the electrophysiological profiles of organoids derived from healthy and diseased individuals. Researchers can identify specific neural dysfunctions associated with various conditions. Drug screening. Electrophysiological assays can be used to evaluate the effects of pharmaceutical compounds on neural activity, aiding in the discovery of new treatments for neurological disorders. Challenges and future directions. Despite its advantages, electrophysiological characterization of 3D printed brain organoids faces several challenges. Technical complexity. Techniques like patch clamp recording are technically demanding and require skilled operators. Integration with 3D structures. Adapting electrophysiological methods to the complex 3D architecture of brain organoids is challenging but essential for accurate measurements. Future directions in this field include the development of more integrated and automated systems for electrophysiological recording, and the combination of electrophysiological data with other modalities, such as imaging and genomic analysis, to provide a comprehensive understanding of neural function in 3D printed brain organoids. 4.4 Applications in neurodevelopmental studies. 3D printed neural networks are used to study the development of the brain, including the formation and maturation of synapses, the development of connectivity, and the effects of genetic mutations on these processes. Studying early brain development. 3D printed brain organoids provide a unique platform to investigate the early stages of brain development. Researchers can observe the differentiation of pluripotent stem cells into various neural cell types, the formation of neural networks, and the establishment of brain-like tissue structures. This allows for a detailed analysis of the molecular and cellular mechanisms underlying neurodevelopment. Modeling neurodevelopmental disorders. Brain organoids can be used to model a range of neurodevelopmental disorders, such as autism spectrum disorder, ASD, schizophrenia, and intellectual disabilities. By creating organoids from patient-derived induced pluripotent stem cells, iPSCs, scientists can study disease-specific cellular behaviors and neural network dysfunctions. This approach helps in understanding the genetic and environmental factors contributing to these disorders. Investigating genetic mutations. The impact of specific genetic mutations on brain development can be examined using 3D printed brain organoids. CRISPR-Cas9 gene editing technology can be employed to introduce mutations into iPSCs, which are then used to generate organoids. Researchers can observe how these mutations affect neural differentiation, network formation, and overall brain structure, providing insights into the etiology of various genetic conditions. Exploring environmental influences. Environmental factors such as toxins, drugs, and nutritional deficiencies can significantly impact brain development. Brain organoids serve as a valuable tool to study these effects. In a controlled environment, researchers can expose organoids to different environmental conditions and analyze the resulting changes in neural development, offering a better understanding of how external factors contribute to neurodevelopmental abnormalities. Drug screening and therapeutic development. One of the key applications of brain organoids in neurodevelopmental studies is drug screening. Organoids can be used to test the efficacy and safety of potential therapeutic compounds in a human-like brain model. This approach helps identify drugs that can correct developmental abnormalities. or alleviate symptoms of neurodevelopmental disorders. Moreover, it provides a more accurate prediction of drug responses, compared to traditional animal models, studying synapse formation and function. Synapse formation is a critical aspect of neurodevelopment. Brain organoids allow for the study of synaptogenesis, the process by which neurons form synaptic connections. Researchers can investigate the factors that promote or inhibit synapse formation, the development of synaptic plasticity, and the mechanisms of synaptic signaling. These studies are essential for understanding how neural circuits are established and how they function in a developing brain. Interrogating neural network activity. Advanced techniques such as multi-electrode arrays, MEAs, and calcium imaging are used to study the electrical activity of neural networks within brain organoids. These methods enable researchers to monitor the firing patterns of neurons, network synchronization, and response to stimuli. By analyzing neural network activity, scientists can gain insights into the functional aspects of brain development and identify abnormalities associated with neurodevelopmental disorders. Personalized neurodevelopmental studies. Personalized medicine approaches benefit greatly from the use of patient-specific brain organoids. By creating organoids from the cells of individuals with neurodevelopmental disorders, researchers can study personalized disease mechanisms and responses to treatments. This personalized approach allows for the identification of tailored therapeutic strategies that are more effective for individual patients. Ethical and practical considerations. While brain organoids provide a powerful tool for neurodevelopmental studies, ethical considerations must be addressed. These include the ethical sourcing of cells, potential implications of creating complex human brain tissue, and ensuring that research practices align with ethical standards. Additionally, practical challenges such as standardizing organoid production and ensuring reproducibility across different laboratories need to be considered. Future directions in neurodevelopmental research. The future of neurodevelopmental research with brain organoids includes integrating more complex cellular and tissue interactions, incorporating vascularization for improved nutrient and waste exchange, and developing more sophisticated imaging and analytical techniques. Ongoing advancements aim to create even more accurate and functional brain models, facilitating deeper insights into brain development and neurodevelopmental disorders. Case studies and applications. A notable example is the work done at the University of Wisconsin-Madison, where scientists have successfully 3D printed brain tissue that can grow and function like typical brain tissue. This tissue exhibits neural activity and has potential applications in drug testing, disease modeling, and understanding neural development. 5.1 University of Wisconsin-Madison study. The University of Wisconsin-Madison has been at the forefront of 3D printed brain tissue research. Their study focused on creating brain organoids that not only resemble the structural aspects of the human brain but also exhibit functional characteristics. The team used induced pluripotent stem cells (iPSCs) to generate neural cells, which were then printed into a 3D structure. Methodology. Cell preparation. iPSCs were differentiated into neural progenitor cells. Bioprinting process. The cells were suspended in a bioink composed of alginate and gelatin, which was printed layer by layer to form the organoid. Maturation. The printed organoids were cultured in a bioreactor to promote cell differentiation and maturation. Results. Neural activity. The organoids exhibited spontaneous neural activity, which was monitored using electrophysiological techniques. Network formation. The neural cells formed synapses and networked similarly. 2. In vivo brain tissue. Applications. These organoids have been used for drug testing, where their response to different compounds has provided insights into potential therapeutic effects and toxicity. 5.2 Harvard University innovations. Researchers at Harvard University have made significant advancements in the vascularization of 3D printed brain organoids. Their work focuses on integrating endothelial cells into the organoids to form functional blood vessels, which is crucial for the long-term viability of the tissue. Methodology. Cell integration. Human endothelial cells were mixed with neural progenitor cells before the printing process. Bioink composition. A composite bioink containing fibrin and collagen was used to support both neural and endothelial cell growth. Vascular network development. The printed constructs were cultured under conditions that promoted angiogenesis and vasculogenesis. Results: vessel formation. The endothelial cells self-organized into capillary-like structures, creating a vascular network within the organoids. Enhanced viability. The presence of blood vessels significantly improved the survival and functionality of the neural cells by providing essential nutrients and removing waste products. Applications: these vascularized organoids are being used to study ischemic brain injuries and neurovascular diseases, providing a more accurate model for these conditions. Additional case studies: Stanford University research project developing brain organoids to study developmental disorders. Methodology using CRISPR-Cas9 to introduce specific genetic mutations into iPSCs. Before differentiation and printing. Results: created organoids that model conditions like autism and epilepsy, providing insights into the genetic and cellular mechanisms underlying these disorders. University of California, San Francisco UCSF. Project investigating the effects of environmental toxins on brain development. Methodology exposing printed brain organoids to various toxins and monitoring their effects on neural development and function. Results: identified specific toxins that disrupt neural network formation and synaptic activity, contributing to our understanding of environmental factors in neurodevelopmental disorders. ETH Zurich. Project engineering brain organoids with integrated sensory input. Methodology incorporating sensory neurons into the organoids and connecting them to external stimuli. Results: created organoids that can respond to light and mechanical stimuli, opening new avenues for studying sensory processing and integration in the brain. 5.3 Industry applications. 3D printed brain organoids are gaining significant traction in the biotechnology and pharmaceutical industries due to their potential to revolutionize drug discovery, personalized medicine, and neurotoxicology testing. Here, we delve into the specific ways these organoids are being utilized across various industry sectors. High-throughput drug screening. The use of 3D printed brain organoids allows for high-throughput screening of pharmaceutical compounds, offering a more physiologically relevant environment than traditional 2D cell cultures. This enables enhanced predictive accuracy. Organoids better mimic human brain tissue, providing more accurate predictions of drug efficacy and toxicity. Reduction in animal testing. By offering an alternative to animal models, organoids help reduce the ethical and logistical burdens associated with animal testing. Cost and time efficiency. Automated systems can rapidly screen thousands of compounds, speeding up the drug discovery process and reducing costs. Personalized medicine. 3D printed brain organoids derived from patient-specific cells enable personalized approaches to treatment. Patient-specific models. Organoids can be created using a patient's own induced pluripotent stem cells iPSCs, allowing for the study of individual disease mechanisms and responses to treatment. Tailored therapeutics. By testing drugs on patient-derived organoids, clinicians can identify the most effective treatments with minimal side effects, leading to more personalized and effective healthcare solutions. Neurotoxicology testing. The neurotoxicity of new compounds can be effectively assessed using brain organoids. Safety profiling. Organoids provide a relevant model for evaluating the neurotoxic effects of drugs, chemicals, and environmental toxins, ensuring safety before clinical trials. Mechanistic insights. Studies on organoids help elucidate the mechanisms of neurotoxicity, aiding in the development of safer compounds and mitigation strategies. Biotechnology innovation. Biotech companies are leveraging 3D printed brain organoids to drive innovation in several areas. Disease modeling. Organoids are used to model various neurological conditions such as Alzheimer's disease, Parkinson's disease, and epilepsy, providing insights into disease progression and potential therapeutic targets. Regenerative medicine. Research into the regenerative potential of brain organoids is paving the way for future treatments aimed at repairing brain damage or restoring lost neural functions. Bioprinting technologies. Companies are continually developing advanced bioprinting techniques and materials, enhancing the fidelity and functionality of printed tissues. Collaborative efforts. Collaborations between industry and academia are crucial for advancing the applications of 3D printed brain organoids. Public-private partnerships joint efforts facilitate the translation of research findings into practical applications, accelerating the development of new treatments and technologies. Consortia and networks. Industry consortia and research networks promote the sharing of knowledge, resources, and technologies, fostering innovation and addressing common challenges. Case studies. Emulate, Inc., a biotechnology company that utilizes organ-on-a-chip technology, including brain organoids, for drug testing and disease modeling. Their brain chip platform provides a dynamic environment for studying brain function and disease in vitro. Organovo, known for its work in bioprinting human tissues, Organovo has been exploring the creation of brain tissues for research and therapeutic purposes, contributing to advancements in neurodegenerative disease modeling. Axosome specializes in nerve-on-a-chip technology, which includes the development of brain organoids for neurotoxicity testing and drug discovery, offering predictive models for human brain responses. Future prospects. The integration of 3D printed brain organoids into industry applications is expected to grow, driven by ongoing research and technological advancements. Enhanced organoid complexity. Future developments aim to increase the complexity and functionality of brain organoids, making them even more representative of human brain tissue. AI and machine learning. The incorporation of artificial intelligence and machine learning algorithms will enhance the analysis of organoid data, improving the predictive power and efficiency of drug screening processes. Expanded applications beyond drug discovery and toxicology. Brain organoids will likely find new applications in neurodevelopmental studies, aging research, and the testing of neuroprosthetics and brain-computer interfaces. In summary, the use of 3D printed brain organoids in industry applications is transforming the landscape of biomedical research and drug development. These organoids offer a powerful tool for high-throughput screening, personalized medicine, neurotoxicology testing, and biotechnological innovation, with promising future directions in enhancing their complexity and expanding their applications. 5.4 Clinical research implications. The use of 3D printed brain organoids in clinical research holds great promise for advancing our understanding of neurological conditions and developing personalized therapeutic strategies. Here are some detailed insights into their clinical research implications. Patient-specific disease modeling. By using patient-derived cells, researchers can create brain organoids that closely replicate the genetic and cellular environment of an individual's brain. This approach allows for the study of patient-specific disease mechanisms and progression. For example, researchers can generate brain organoids from patients with genetic mutations linked to conditions like Alzheimer's disease or autism, providing a unique platform for investigating how these mutations affect neural development and function. Personalized drug testing. 3D printed brain organoids enable personalized drug testing by allowing researchers to test the efficacy and safety of potential treatments on patient-specific tissues. This can help identify the most effective therapies for individual patients, minimizing adverse reactions and improving treatment outcomes. Personalized drug testing can accelerate the development of targeted therapies for complex neurological disorders. Understanding disease heterogeneity. Neurological disorders often exhibit significant heterogeneity, with different patients experiencing varied symptoms and disease trajectories. Brain organoids can help researchers explore this variability by providing a diverse range of disease models. This can lead to a better understanding of the underlying mechanisms that contribute to disease heterogeneity and inform the development of more inclusive and effective treatment strategies. Early detection and intervention. Brain organoids can be used to study early disease stages and identify biomarkers for early detection. By analyzing how disease processes unfold in organoids, researchers can discover early indicators of disease onset, potentially leading to earlier diagnosis and intervention. This is particularly important for neurodegenerative diseases, where early treatment can significantly impact disease progression and patient quality of life. Ethical and regulatory considerations. The use of 3D printed brain organoids in clinical research must adhere to ethical guidelines and regulatory standards. This includes ensuring informed consent for the use of patient-derived cells, protecting patient privacy, and meeting regulatory requirements for clinical applications. Addressing these considerations is crucial for the responsible integration of this technology into clinical practice. Potential for regenerative medicine. In addition to disease modeling and drug testing, 3D printed brain organoids hold potential for regenerative medicine. Researchers are exploring the possibility of using these organoids to develop cell-based therapies for repairing brain damage caused by injury or disease. This could pave the way for innovative treatments that restore lost neural functions and improve patient outcomes. Collaboration with healthcare providers. Effective clinical research with 3D printed brain organoids requires collaboration between researchers, healthcare providers, and patients. By working together, these stakeholders can ensure that research is aligned with clinical needs, and that new discoveries are rapidly translated into clinical applications. This collaborative approach can enhance the impact of research and lead to more effective and patient-centered healthcare solutions. In summary, the clinical research implications of 3D printed brain organoids are vast and transformative. These organoids offer a powerful tool for understanding neurological diseases, personalizing treatment, and exploring new therapeutic avenues. As research advances, the integration of brain organoids into clinical practice holds the potential to revolutionize the diagnosis, treatment, and management of neurological disorders. Ethical considerations and regulatory challenges. The use of 3D printed brain tissues in research and medicine raises ethical and regulatory challenges. These include ensuring the ethical sourcing of cells, maintaining patient privacy in personalized medicine applications, and navigating the regulatory landscape for clinical use. Addressing these challenges is essential for the responsible advancement of this technology. 6.1 Ethical sourcing of cells. The origin of cells used in 3D printing must be ethically sourced, with informed consent from donors and adherence to ethical guidelines. 6.2 Privacy concerns. In personalized medicine, the use of patient-derived cells necessitates stringent measures to protect patient privacy and data security. 6.3 Regulatory approvals. Navigating the regulatory landscape for the clinical use of 3D printed tissues involves meeting rigorous standards for safety, efficacy, and manufacturing practices. 6.4 Societal impact. The societal implications of 3D printed brain tissues include potential changes in how diseases are treated, the accessibility of advanced therapies, and the ethical considerations of creating complex human tissues in vitro. Future directions in research and development. Future research directions include the development of more sophisticated bioprinting technologies, exploration of new biomaterials, and interdisciplinary collaborations. Advancements in imaging techniques will also play a crucial role in monitoring tissue development and functionality. These efforts aim to enhance the precision and scalability of 3D printed brain organoids. 7.1 Next-generation bioprinters. The development of next-generation bioprinters with higher resolution and multi-material capabilities will enable the creation of more complex and functional brain tissues. An introduction to next-generation bioprinters. Next-generation bioprinters represent a significant leap forward in the field of tissue engineering and regenerative medicine. These advanced devices are designed to overcome the limitations of traditional bioprinters by offering higher precision, multi-material capabilities, and enhanced control over the microenvironment of printed tissues. Precision and resolution. One of the key features of next-generation bioprinters is their ability to print with extremely high precision and resolution. This is achieved through advancements in nozzle design, printing techniques, and software algorithms. High-resolution printing is crucial for replicating the intricate structures of brain tissue, including neural networks and blood vessels. Multi-material printing. Next-generation bioprinters can simultaneously print multiple biomaterials, allowing for the creation of more complex and functional tissue constructs. This capability is essential for mimicking the heterogeneous composition of brain tissue, which includes various cell types, extracellular matrix components, and supporting structures. Advanced bioinks. The development of advanced bioinks is a critical component of next-generation bioprinting. These bioinks are formulated to provide optimal conditions for cell viability, proliferation, and differentiation. They can be customized to include specific growth factors, signaling molecules, and structural proteins that mimic the natural brain environment. Integration with imaging technologies. Next-generation bioprinters are often integrated with advanced imaging technologies, such as live cell imaging, high-resolution microscopy, and real-time monitoring systems. This integration allows for precise control and validation of the printing process, ensuring that the printed tissues meet the desired specifications. Automation and scalability. Automation is another key feature of next-generation bioprinters. Automated systems can handle complex printing tasks with minimal human intervention, increasing reproducibility and throughput. Scalability is also enhanced, allowing for the production of larger tissue constructs and the potential for industrial-scale manufacturing. Examples of next-generation bioprinters. Several next-generation bioprinters have been developed by leading research institutions and biotechnology companies. BioX6 Bioprinter by CELLINK. This printer offers 6 printheads for multi-material printing in high precision, suitable for creating complex tissue models. Regenova Bioprinter by Cyfuse Biomedical uses a unique needle array system for scaffold-free tissue construction, enabling high-resolution printing of intricate structures. Light-based bioprinter by Prellis Biologics utilizes holographic bioprinting technology to achieve submicron resolution, ideal for vascularization and detailed tissue engineering. Applications in brain organoid research. The capabilities of next-generation bioprinters are particularly valuable in brain organoid research. They enable the creation of highly detailed brain models that can replicate the cellular architecture and functionality of the human brain. Applications include neurodevelopmental studies investigating the formation and maturation of neural networks, disease modeling creating patient-specific models for studying neurological diseases, such as Alzheimer's, Parkinson's, and autism, drug testing providing accurate platforms for high-throughput screening of neuroactive compounds, personalized medicine developing tailored therapies based on individual patients' cellular characteristics. In future directions and innovations. The future of next-generation bioprinters is focused on further enhancing precision, incorporating new biomaterials, and integrating more sophisticated monitoring and control systems. Innovations may include hybrid printing techniques combining different printing methods to achieve unprecedented levels of detail and functionality. Smart bioinks developing bioinks that can respond to environmental cues and dynamically adjust their properties. Artificial intelligence integration using AI to optimize printing parameters and predict tissue development outcomes. Miniaturization and portability creating compact, portable bioprinters for on-site medical applications and research in remote locations. By continuing to advance the capabilities of next-generation bioprinters, researchers and engineers are paving the way for breakthroughs in tissue engineering, regenerative medicine, and personalized healthcare. 7.2 Innovative biomaterials. Innovative biomaterials play a critical role in the development of 3D printed brain organoids. These materials must mimic the natural environment of brain tissues, supporting cell growth, differentiation, and the formation of functional neural networks and vascular structures. The selection and engineering of these biomaterials are pivotal for the success of bioprinted neural tissues. Types of biomaterials. Hydrogels. Hydrogels are widely used due to their high water content and biocompatibility, closely resembling the extracellular matrix ECM of brain tissues. Common hydrogels include alginate derived from seaweed. Alginate is biocompatible and forms gels in the presence of divalent cations like calcium. It is often used in combination with other materials to enhance its properties. Gelatin, a denatured form of collagen, gelatin, is biodegradable and supports cell adhesion and proliferation. It can be cross-linked to form stable gels. Collagen as a major component of the natural ECM. Collagen provides excellent biocompatibility and supports cell growth and differentiation. Synthetic polymers. Synthetic polymers offer tunable mechanical properties and degradation rates. Common synthetic polymers include polyethylene glycol PEG is biocompatible and can be functionalized to incorporate bioactive molecules, providing a customizable scaffold. Polylactic acid PLA and polyglycolic acid PGA. These biodegradable polymers are used in various tissue engineering applications, including neural tissue scaffolding. Hybrid materials. Hybrid materials combine natural and synthetic polymers to leverage the advantages of both. These materials can be engineered to have specific mechanical and biological properties tailored to the requirements of neural tissue printing. Functionalization of biomaterials. Bioactive molecules. Incorporating bioactive molecules such as growth factors, peptides, and proteins into biomaterials can enhance cell adhesion, proliferation, and differentiation. Commonly used bioactive molecules include neurotrophic factors such as brain-derived neurotrophic factor BDNF and nerve growth factor NGF, which support the survival and growth of neurons. Extracellular matrix proteins such as laminin and fibronectin, which promote cell adhesion and differentiation. Nanoparticles. Nanoparticles can be embedded within biomaterials to provide additional functionalities such as controlled drug release. Nanoparticles can be loaded with drugs or growth factors and released in a controlled manner to enhance tissue development. Electrical conductivity. Incorporating conductive nanoparticles can enhance the electrical properties of the printed neural tissues, improving their functionality. Advanced techniques in biomaterial engineering. Bioprinting strategies. Advanced bioprinting strategies include the use of multimaterial printing, combining different biomaterials within a single print to create more complex and functional tissues. Gradient printing, creating gradients of biomaterials and bioactive molecules to mimic the natural variations found in tissues. Dynamic scaffolds. Dynamic scaffolds are designed to change their properties in response to environmental cues, such as pH-responsive materials. These materials change their structure or properties in response to pH changes, allowing for targeted release of bioactive molecules. Temperature-responsive materials. Materials that gel or liquefy in response to temperature changes, facilitating cell encapsulation and scaffold formation. Challenges and future directions. Compatibility and degradation. Ensuring the biocompatibility and appropriate degradation rates of biomaterials is crucial. Future research aims to develop materials that degrade at rates matching tissue regeneration. Mechanical properties. Balancing the mechanical properties of biomaterials to match those of native brain tissue, while supporting cell function, is a key challenge. Innovative materials with tunable stiffness and elasticity are being explored. Customization and scalability. Developing customizable and scalable biomaterials is essential for widespread application. Research focuses on creating materials that can be easily modified to meet specific research and clinical needs. Integration with living systems. Future biomaterials will need to integrate seamlessly with living systems, supporting not only cell growth and differentiation, but also functional integration with host tissues in therapeutic applications. Conclusion. Innovative biomaterials are fundamental to the success of 3D printed brain organoids. The ongoing development and refinement of these materials will continue to enhance the capabilities and applications of bioprinted neural tissues, driving advances in biomedical research and therapeutic strategies. 7.3 Interdisciplinary collaborations. Interdisciplinary collaborations are essential for the advancement of 3D printed brain organoids. These partnerships bring together expertise from various fields, fostering innovation and addressing the complex challenges associated with creating functional brain tissues. Here's a detailed look at the different aspects of interdisciplinary collaborations. Importance of collaboration. Interdisciplinary collaborations are vital for integrating knowledge, combining insights from biology, engineering, materials science, and computer science to develop sophisticated bioprinting techniques and materials. Innovative solutions, encouraging creative problem solving by leveraging diverse perspectives and expertise. efficiency and speed accelerating the pace of research and development. Through shared resources and collaborative efforts. Key disciplines involved. Biology and neuroscience understanding the cellular and molecular mechanisms of brain development. Neural differentiation and network formation. Materials science developing advanced biomaterials that mimic the brain's extracellular matrix and support cell growth and differentiation. Biomedical engineering designing and optimizing bioprinting. Technologies and devices for precise and reproducible tissue fabrication. Computer science and bioinformatics utilizing computational modeling, machine learning, and data analysis to optimize printing protocols and predict tissue behavior. Clinical medicine translating research findings into clinical applications, ensuring that the developed tissues meet medical standards and addressing patient-specific needs. Examples of collaborative projects. The NIH Brain Initiative, a collaboration involving neuroscientists, engineers, and clinicians aimed at mapping the human brain and developing new technologies for brain research. European Union's Human Brain Project integrates efforts from neuroscience, medicine, and computing to create detailed models of the brain, with applications in understanding brain function and disease. Harvard Wyss Institute, known for its interdisciplinary approach, combining biology, engineering, and material science to create innovative 3D bioprinting solutions, including vascularized brain organoids, or collaborative research centers and institutes. The Wyss Institute for Biologically Inspired Engineering focuses on bio-inspired materials and devices, fostering collaborations across biology, engineering, and medicine. MIT Media Lab integrates disciplines like computer science, neuroscience, and engineering to push the boundaries of 3D printing and bioprinting technologies. Stanford BioX, an interdisciplinary initiative combining biology, engineering, and medicine to advance biomedical research, including brain organoid development. Challenges in interdisciplinary collaborations. Communication barriers. Differences in terminology and methodologies across disciplines can hinder effective communication and collaboration. Resource allocation. Coordinating funding, infrastructure, and human resources among different departments and institutions can be challenging. Intellectual property. Managing intellectual property rights and sharing benefits equitably among collaborators can be complex. Cultural differences. Academic, industrial, and clinical partners often have different priorities and working cultures, which can affect collaboration dynamics. Strategies for successful collaboration. Establishing clear objectives. Defining common goals and expectations from the outset to align efforts and resources. Regular communication. Maintaining open and frequent communication through meetings, workshops, and collaborative platforms. Shared resources. Creating centralized facilities and databases to share tools, materials, and data among collaborators. Interdisciplinary training. Promoting cross-disciplinary education and training programs to build a workforce capable of bridging gaps between fields. Future directions. Enhanced collaboration platforms. Developing digital platforms for better coordination and data sharing among interdisciplinary teams. Global collaborations. Expanding collaborations beyond national borders to include international expertise and perspectives. Public-private partnerships. Encouraging partnerships between academic institutions, industry, and government agencies to drive innovation and commercialization of 3D printed brain organoid technologies. Patient involvement. Integrating patient input into research to ensure that developed technologies meet real-world clinical needs and ethical standards. Interdisciplinary collaborations are the cornerstone of progress in 3D printed brain organoid technology, driving innovation and overcoming the multifaceted challenges in this emerging field. 7.4 Imaging and monitoring technologies. The advancement of imaging and monitoring technologies plays a crucial role in the development and evaluation of 3D printed brain organoids. These technologies enable researchers to visualize, track, and assess the structural and functional properties of organoids in real time, providing critical insights into their development and performance. Live cell imaging. Live cell imaging is an essential technique for observing the dynamic processes within 3D printed brain organoids. It allows for the real-time visualization of cell behavior, including migration, proliferation, and differentiation. Advanced microscopy techniques, such as confocal and multiphoton microscopy, are commonly used to capture high-resolution images of live tissues. High-resolution microscopy. High-resolution microscopy techniques, including super-resolution microscopy e.g., STED, SIM and electron microscopy, provide detailed images of the cellular and subcellular structures within brain organoids. These techniques are critical for examining the intricate architecture of neural networks and the formation of synapses. Functional magnetic resonance imaging fMRI. Functional MRI fMRI is a non-invasive imaging technique used to measure and map brain activity. In the context of 3D printed brain organoids, fMRI can be adapted to monitor the functional connectivity and activity patterns within the organoids, providing insights into their neural functionality. Calcium imaging. Calcium imaging is a widely used technique to monitor neuronal activity. It involves the use of calcium-sensitive fluorescent dyes, or genetically encoded calcium indicators to visualize changes in calcium levels within neurons, which correspond to neural activity. This technique is valuable for studying the functional properties of neural networks in brain organoids. Electrophysiological monitoring. Electrophysiological techniques, such as patch clamp recording and multi-electrode arrays (MEAs), are used to measure the electrical activity of neurons within 3D printed brain organoids. These methods provide detailed information on the functional connectivity and excitability of neural networks, allowing researchers to assess the physiological relevance of the organoids. Optical coherence tomography, a non-invasive imaging technique that provides high-resolution, cross-sectional images of tissues. In the context of brain organoids, October can be used to monitor tissue architecture and vascularization over time, aiding in the assessment of structural integrity and development. Advanced imaging software. Advanced imaging software and computational tools are essential for analyzing and interpreting the vast amounts of data generated by imaging techniques. These tools enable the reconstruction of 3D images, quantitative analysis of cellular and network properties, and modeling of neural dynamics. Integration of imaging and bioprinting. The integration of imaging technologies with bioprinting processes allows for real-time monitoring and quality control during the fabrication of brain organoids. This integration ensures that the printed tissues meet the desired structural and functional specifications, enhancing the reproducibility and reliability of the organoids. By leveraging these advanced imaging and monitoring technologies, researchers can gain comprehensive insights into the development, structure, and function of 3D printed brain organoids. These technologies not only facilitate the optimization of bioprinting processes, but also contribute to the broader understanding of brain development and disease mechanisms. Comparison with traditional methods. Compared to traditional methods of neural tissue engineering, 3D printing offers several advantages, including higher precision, reproducibility, and the ability to create more complex tissue structures. However, it also presents challenges such as ensuring the long-term viability of printed tissues and achieving the precise replication of complex brain structures. 8.1 Advantages of 3D printing. Precision and reproducibility. 3D printing allows for precise control over cell placement and tissue architecture, leading to highly reproducible results. Traditional methods, which often rely on manual techniques, lack this level of precision, resulting in variability between samples. Complexity and customization. The ability to create intricate tissue structures that closely mimic the natural brain environment is a significant advantage of 3D printing. This includes the replication of complex neural networks and the incorporation of multiple cell types in precise spatial arrangements. High throughput capability. 3D printing enables high-throughput production of brain organoids, facilitating large-scale studies and drug screening processes. Traditional methods are more time-consuming and labor-intensive, limiting their scalability. Integration of multiple cell types. 3D printing can simultaneously integrate various cell types, including neurons, glial cells, and vascular cells, into a single construct. Traditional methods often require separate steps to combine these cell types, which can be less efficient and more prone to errors. 8.2 Challenges and limitations. 1. Viability and longevity. Ensuring the long-term Viability of printed tissues remains a significant challenge. Printed tissues must maintain their structural integrity and functionality over extended periods, which is crucial for applications in disease modeling and drug testing. Complex replication. Replicating the complex architecture and functionality of natural brain tissues requires ongoing innovation. Traditional methods often involve simpler, two-dimensional cultures, which cannot fully replicate the 3D structure and dynamic environment of the brain. Standardization. Achieving standardization in 3D printing processes is challenging due to the variability in printer performance, biomaterials, and cell sources. Traditional methods, while less precise, have more established protocols and standards. Cost and accessibility. The initial cost of 3D bioprinters and biomaterials can be high, potentially limiting accessibility for some research institutions. Traditional methods, which typically use more affordable equipment and materials, may be more accessible, but offer less advanced capabilities. 8.3 Comparative studies. Structural fidelity. Studies comparing traditional tissue engineering methods with 3D printing have demonstrated the superior structural fidelity of 3D printed brain organoids. These organoids better replicate the intricate architecture of the brain, including the formation of neural networks and vascular structures. Functional outcomes. 3D printed tissues have shown enhanced functional outcomes, such as improved electrical activity and synaptic connectivity, compared to traditional methods. These functional improvements are crucial for studying neural behavior and disease mechanisms. Applications in drug testing. In drug testing applications, 3D printed brain organoids provide more accurate predictions of drug efficacy and toxicity compared to traditional 2D cultures. This is due to their ability to mimic the complex interactions within the brain environment. 8.4 Future improvements. Advancements in bioprinting technology. Continued advancements in bioprinting technology, including higher resolution printers and more versatile printing techniques, are expected to further enhance the capabilities of 3D printed brain tissues. Development of new biomaterials. Exploration of new biomaterials that better mimic the brain's extracellular matrix and promote cell differentiation and network formation is a key area of research. These materials will improve the viability and functionality of printed tissue. Integration with advanced imaging techniques. Integrating 3D printed brain organoids with advanced imaging techniques, such as live cell imaging and high-resolution microscopy, will enhance our ability to monitor tissue development and functionality in real time, leading to better insights and refinement. Interdisciplinary collaboration. Collaborations between biologists, engineers, and material scientists will drive innovation in 3D printed brain organoids. These interdisciplinary efforts are essential for overcoming current technical challenges and expanding the applications of this technology. 8.5 Summary. Compared to traditional neural tissue engineering methods, 3D printing offers higher precision, reproducibility, and the ability to create complex structures. Advantages include detailed control over cell placement, integration of multiple cell types, and high throughput capabilities. However, challenges such as ensuring long-term viability, achieving complex replication, and high costs persist. Studies have shown that 3D printing provides superior structural fidelity and functional outcomes. Future improvements in bioprinting technology, new biomaterials, advanced imaging integration, and interdisciplinary collaboration are expected to enhance the capabilities and applications of 3D printed brain tissues. Conclusion. 3D printing technologies for brain organoids are advancing rapidly, with significant progress in creating functional neural networks and blood vessels. These innovations hold great promise for biomedical research and therapeutic applications, providing new tools for understanding and treating neurological disorders. 9.1 Summary of key points. Advancements. Rapid advancements in 3D printing and bioprinting techniques. Applications. Significant applications in research, drug development, and personalized medicine. Challenges. Ethical and regulatory challenges that need to be addressed. 9.2 Impact on neurological research. The creation of functional brain organoids is revolutionizing neurological research by providing more accurate models for studying brain function and disease. 9.3 Therapeutic potential. The potential for developing new therapies for neurological disorders through the use of 3D printed brain tissues is immense, offering hope for more effective treatments. 9.4 Future Prospects. The future of 3D printed brain organoids looks promising, with ongoing research focused on overcoming current limitations and exploring new applications. 10. Conclusion. The development of 3D printing technologies for brain organoids, with functional neural networks and blood vessels, is a major breakthrough in biomedical research. This technology enables the creation of complex brain tissue models that mimic the natural brain environment, facilitating neural development studies, disease modeling, and drug testing. The key areas of focus include bioprinting techniques, advanced vascularization methods, and the integration of neural networks. Ethical considerations and regulatory challenges are crucial for the responsible advancement of this technology. Future research aims to enhance the precision and scalability of 3D printed brain organoids, offering significant potential for personalized medicine and therapeutic applications.